So, welcome back to the lecture series on biosecurity measures and specifically the development of biosensor. So, last week we talked about the production of antibodies by different methods using hybridoma methods and other methods, and we extensively studied about the fundamentals of using molecular techniques to generate antibodies, and these antibodies are essentially one of the most potent tool for biosensor development. And I highlighted the fact that next generation of biodesigners working in this area of biosensor development and design will have to have a comprehensive understanding of biology, chemical engineering, physics, engineering, micromachining, microfabrication, microfluidics, microelectromechanical systems, and packaging, and integrating this whole thing using different kind of softwares. So, today one such breakthrough we will be discussing, which is lateral flow immunoassays. You could see in the picture where in the circle you could see this is where the lateral flow immunoassays stands, and today we will talk about this particular technique, which has helped us to develop wide array of detection tools using different kind of base materials. They are even flexible nowadays. Initially, they were much more solid matrix. But before I start lateral flow immunoassays, one of the major advancement which has happened in last 20 to 30 years is micromachining and microfluidics and biomems. These are three areas where it has been possible to create microstructures on different kind of substrates, and these microstructures have been used to develop different kind of flow cells, microchannels, and since micromachining has come of an age today, it is possible to integrate a lot of optical devices into these micromachined structures. So, in a way, the development of microelectronics, micromachining, microfabrication has completely revolutionized the whole field of biosensor development. Otherwise, the logics are still the same. There has to be some antibody-antigen reaction, there has to be something against a toxin which will bind, but the real challenge comes, as I have mentioned earlier multiple times, is how you distinguish between the noise and a signal, because these are molecular interactions. There are conjugated secondary antibodies or fluorescent probes or nano probes which are used for detecting signals. So much of the advancement in this field, we owe it to microfluidics and microfabrication and biomems technologies. So this is where we are today. So we will be talking about signal generation devices, which is LFIA, lateral flow immunoassay. As the name indicates, it's an immunoassay. It means there has to be antigen antibody reactions. Okay. So if we talk about, so today's class title will be design and fabrication of lateral flow immunoassays. So if we talk about immunoassays, LFIAs, these are quick, simple and cheap assays to analyze various samples at the point of care or in the field, making them one of the most widespread biosensors currently available. They have been successfully employed for detection of myriad of different targets ranging from atoms up to whole cells in all types of samples including water, blood, foodstuff, environmental sample, urine and other feces. These lateral flow immunoassays are being used. So, it is a very potentially effective tool for field application, for transporting them at distance, for using them for our day-to-day detection to extremely tricky and tough detection. So, LFIA, if we look at it, the operation relies on capillary work of samples throughout a series of sequential pads, each with different functionalities, aiming to generate a signal to indicate the absence or presence, and in some cases, the concentration of the analyte of interest. To have user-friendly operations, their development requires the optimization of multiple interconnected parameters that may overwhelm the new developers. As I was mentioning, LFIA development is a challenge for microfluidics, biomems, and optics, and microelectronics, because much of these assays relies heavily on engineering these devices. Now, if we look at the schematic of the main component and operation of a typical LFA, an LFA strip is made up of four main parts: the sample pad, the conjugate pad, the membrane, and the absorbent pad. They are mounted on a laminated card. The membrane binds to the capture bioreceptors that form the test and control lines. So, here you see the sample pad, you have the conjugate pad, detection of bioreceptor nanoparticle conjugate, and here you have the test line, okay, to capture bioreceptor, and a control line to capture bioreceptor. Here you have the adsorbent pad, laminated card, but you do not see any of the detection units, okay. They are mounted on laminated card. The membrane binds to the capture bioreceptor that forms the test and control lines. Panel B and C, you could follow, this is the B and this is the panel C, shows the operation of an LFA based on immunosandwich recognition. In the B, the presence of target analyte in the sample results in accumulation of the nanoparticle on the test and control lines, making the classical two red line appears, as you could see out here. In the absence of the target analyte, you could see here, the nanoparticle accumulate only on the control line, giving it a single red line. So, this is a typical, very most simplistic version of our LFIA assay. Now, think of it when these analytes have to move along. So, the more you make miniaturized it, the more microfabrication is involved in this kind of assay. So, it has to flow. So, it's almost like a flow cell where you have adsorbent on which the analyte is going to bind. So, essentially what you are doing, you have a surface on which you have very specific binders sitting there. Now, you flow your sample on top of that. If your sample has a particular antigen which is supposed to bind to that antibody, it will bind. The soon it binds, this particular antibody which is adsorbed on this surface has certain marker. It could be a fluorescent marker, it could be a dye-based marker, it could be some other signal. That signal has to be captured once the binding happens. But then you have to ensure that it flows uniformly on the surface of these antigens, of the antibodies which are present on the surface. So, the reaction has to happen in a very particular way. So, you realize that when you are creating these kind of small biochips or biosensors, the amount of precision what is needed is gigantic, because these are used for commercial purpose. In a commercial scale, you cannot afford to have anomalies, or you cannot afford to have two products varying differently from the same company. So it has to be very precise, well measured, and well manufactured, so that the user at the other end does not find ambiguity between two of your sensors. Okay. So now if we look at, generally use one of the following two main sensing strategies. One is a competitive binding strategy, or sandwich or non-competitive assays. Their successful use in LFAs requires the developer to consider the important factor during the sensor design, fabrication, and optimization. So there are multiple ways how the binding can happen. As I mentioned, there is a competitive or sandwich or non-competitive binding assays. What are these different assays, competitive and non-competitive binding assays? Now, when we talk about competitive assays, these are two main types of competitive format. In the first type, the target in the sample and the labeled target, alternatively a molecule with less affinity for bioreceptor than the target itself in the conjugate pad, compete for test line capture bioreceptor. So it is almost, there are two main types of competitive format. In the first type, the target in the sample and a labeled target, affirmatively a molecule with less affinity for the bioreceptor than the target itself in the conjugate pad, compete for test line capture bioreceptor. In the second type, the target in the sample and the target on the test line, which would act as a test line capture bioreceptor, compete for the labeled bioreceptor. It is just the reverse format what are being used for competitive assay and different kind of competitive assays. So basically it is a competitive binding, something which binds to the target better versus something which binds to the target weaker. Based on these binding assays, we decide that how much molecule of interest or pathogen is present on the substrate or on the material what you are sampling. Now, if we look at the competitive assays further, the key characteristic of competitive assay is that higher target concentration result in lower signal. Competitive assays are generally used for small target molecules, which is less than 1 kilodalton, that cannot be efficiently recognized by more than one bioreceptor, such as drug or toxin, but they could also be adapted for big analytes. Since competitive assays are not affected by the hook effect, the detection and capture bioreceptors have all their binding sites occupied by single target, preventing the sandwich formation. Okay. This would show a low signal, which may be misinterpreted as absence or a low target concentration. They are particularly useful for detection of targets with extremely high concentrations. It is important for the developer of competitive assay to verify that the bioreceptor can recognize the competitor molecule even after it is labeled or adsorbed on the membrane. The use of spacer to anchor and the competitor to the membrane or label may expose it and favor its recognition by the bioreceptors. So this is the basic of the competitive assay. Now, if we look at competitive assay further, during the optimization of competitive assays, the concentration of the detection bioreceptor and the test line capture bioreceptor, generally between 1 to 0.1 milligram per ml, are carefully adjusted to not oversaturate the signal in the absence of target, which would provide a low sensitivity LFA. So you have to realize that every system has a detection limit, and you cannot oversaturate the detection limits, because then you are going to get results which are ambiguous, which will confuse you. So you have to ensure that these bindings, concentration you are using, should be within the permissible limits of the binding assays. Okay. So in order to achieve that, what you have to do is what we call as you have to have the initially doing test curves has to be developed. In other words, these test curves will take care of the basic binding kinetics. Once these test curves have been taken care, then it becomes easier to do the detection. Without that, detecting any of these things could be challenging. So you should have an idea about known antigen and antibody reaction. Without the known amount, it is tough to work with these kind of systems. So once again, if we go through the basics of the competitive assays, there are two main types of competitive formats. In the first type, the target in the sample and a labeled target, alternatively molecule with less affinity for the bioreceptor than the target itself, in the conjugate pad compete for the test line capture bioreceptors. So in other words, what is happening is that, say for example, I have this antibody out here. Now I have one binder here, but this is not my target. This binder is kind of a weak binding on substance, but it does bind. Now say for example, somebody more powerful comes which can bind stronger to that. What this will do, it will remove this binder and it will try to bind. Say for example, this is that new binder. This is going to come and dismantle this other binder and will bind more strongly. So that's when we are talking about a competitive assay. There's a competition. So you are pushing your antigen or the toxin to competitively bind on the test line. So that means on the test line, you already have another agent which is a weak binder. As I mentioned here, there are two main types of competitive format. In the first type, the target in the sample and the labeled target, alternatively a molecule with less affinity for the bioreceptor than the target itself. This part is of key importance. Something which has a lesser affinity. So when the one with more affinity will come at that particular site, that's going to dismantle or dislodge that other molecule and will bind on it. Is it making sense? So this is the part what is of extreme understanding that what is happening. So essentially on the test line, you already have a binder present. There are binders present there. So you have the antibody, on top of that there are antigens which are there. But these antigens are weak binders. So now when the flow is coming like this, what this will do is will topple all these weak binders and it will bind on top of it. It is almost like the people who are holding hands have a weaker holding, but the newer ones which are coming of your choice have a stronger anchor. So they will hold it tightly and they will dismantle them from the site. And this is what we call as the competition. There's a competition between the molecules to bind to the target. And this is the fundamental basis of LFIA's competitive assay. So if you look at the slide, once again, in the first type, the target in the sample and a labeled target, alternatively a molecule with less affinity for the bioreceptor than the target itself, in the conjugate pad compete for the test line capture bioreceptor. In the second type, the target in the sample and the target on the test line, which would act as a test line capture bioreceptor, compete for labeled bioreceptors. So if you look at it in this picture, so this is your positive sample and here you have the test lines which are present. So in the test line, what you'll be observing is a competitive binding. This competitive binding is of immense importance to rule out the possibility that this is some kind of unspecific binding or something, because that competitive binding. So, what will happen once the competitive binding will happen? This will dislodge the weak binders. The weak binders will move further ahead. So, that is why it is of extreme importance that we have a positive control and a negative control in all these kind of tests, so that we kind of get a clear understanding about how these binding assays are being evaluated, are being quantified, without getting into the aspect what I was mentioning out here of oversaturation. This oversaturation aspect has to be avoided at any cost. Thank you, and we'll move on to the next class after this.